Combination Th. Morgan experimental verification of chromosomal theory. So his student Alfred Stewart Watt used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between the genes and map their position on the chromosome. Today, genetic maps used in human genome sequencing project. Now, Morgan studied on X chromosome. What he saw that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Okay, now and Morgan and his group found that even when genes were grouped on the same chromosome, some genes were tightly linked, showed very low recombination, while others were loosely linked, showed high recombination. Polygenic inheritance examples human skin color and human height. Such traits are generally controlled by three or more genes and are thus called as polygenic traits. The genotype with all the dominant alleles capital A, capital A, capital B, capital B, capital C will have the darkest skin color, with all the recessive alleles will have the lightest skin color, and in intermediate, like three dominant alleles and three recessive alleles will have an intermediate skin color. Now pleiotropy. A single gene can exhibit multiple phenotypic expression, external expression, right? Multiple. Now such a gene is pleiotropic gene that causing the pleiotropy. Now in pleiotropy affects metabolic pathways. Now the disease is caused by mutation in the gene that codes for the enzyme phenylalanine dehydroxylase, single gene mutation, phenylalanine hydroxylase. Now this manifests itself through phenotypic expression characterized by mental retardation, a reduction in hair and skin pigmentation. So this was pleiotropy. A single gene can exhibit multiple phenotypic expression.